Hello. So today we will be talking about immobilized enzymes. So far we have seen the structure and function of enzymes as they are available in the living cells, and we have also seen that functionally they are very efficient catalysts in the sense that they provide a very high degree of specificity, a very high degree of turnover number as far as their function is concerned. They also can catalyze a number of stereospecific chemical transformations that are otherwise difficult to carry out in a single step by other chemical routes, and also a major advantage of mild operating conditions which make them very attractive industrial catalysts. But the fact is that these catalysts have not found such a widespread use as far as industry is concerned, excepting in some conventional applications in foods or pharmaceuticals, which are the natural processing agent in the case of industrial applications. Bulk of the other industries have not made use of these biocatalysts, at least up to the beginning of last 2 decades. If you look at the industrial applications, I think until 60s there was not a very significant application of enzymes in industrial processing. Now basically the major sort of limitations we also noticed during our understanding of the structure function relationship of the enzyme, that they are usually very unstable, and that probably is one of the major limitations as far as enzymes are concerned with respect to their application. And they cannot be used conventionally in organic solvent. That is the perception. That means their structural conformation in which they are active, catalytically active, is functional only in aqueous medium, and their activity or their function in the normal temperature range also puts sometimes a disadvantageous situation, in the sense that at those temperature many of the microbial activities are also optimum, and therefore they lead to contamination of products, and therefore their industrial application becomes restricted. Now also a major factor in the limitations of the application of soluble enzymes in industry has also been the nature of or rather the soluble nature of the enzymes. That means they have to be used in water soluble form. When we have to use them in water soluble form, it only means that the process has to be in a batch mode. That means you take an enzyme solution or a substrate solution, add a quantity of soluble enzyme in a batch mode, provide the optimum temperature and pH, carry out the reaction, and terminate the reaction by changing one of the operational parameter, either by heating it or by drastic change of pH, so that the reaction is terminated, and then extract the product. Now in this whole process, what we are looking at, one of the major problem is that one has to discard the enzyme, one has to deactivate the enzyme, and the enzyme is usually not possible to be recovered at the end of the process, because recovering the enzyme from the reaction mixture will be as difficult or as tedious, if not more, as production of the original enzyme itself. And in most cases enzymes being produced by living systems are usually expensive commodities, and therefore their deactivation at the end of one batch operation is not a desirable feature, and that has been a major limitation. And of course the two issues of instability and possibility of their use repeatedly or in a continuous mode has been the major driving force of looking at immobilization as an approach to provide a better platform for the use of enzyme in industrial application. Now I think a major question will come that what do we mean by immobilization? What is immobilization? If you look at for a very broad based and operational definition of immobilization, we can say that enzymes physically confined or localized in a defined region of a space. That is essentially as a definition of immobilized enzymes. The enzymes that are physically confined or localized in a defined region of a space with retention of their catalytic activity and which can be used repeatedly or continuously in a process are usually considered as the immobilized enzyme preparations. Now this is basically an operational definition. Now I would like you to concentrate on at least three major phrases built in this definition. One is physically confined or localized in a defined region of space. That means the enzyme molecule which is homogeneously suspended in a soluble preparation has to be then localized in a narrow region of space and is no longer uniformly homogeneously distributed in a reaction mixture. I think an analogy can be considered to a heterogeneous catalysis. I mean, in the case of chemical catalysis, now many of the catalysts are used in the heterogeneous phase, so that instead of having, while the soluble enzyme use of soluble enzyme will provide a homogeneous catalysis, that means the distribution or the composition of every species in the reaction mixture, whether it is catalyst or a substrate or a product, is uniform throughout the reaction mixture. As against that, the use of immobilized enzymes will lead to a heterogeneous catalysis. That means while substrate and the product might be homogeneously distributed, but the enzyme will be localized in some area of space. It will not be homogeneously distributed at every point in the reaction mixture. Then I think the second phrase which is very important is the retention of their catalytic activity. That means if you do any modification so as to arrive at an immobilized enzyme preparation, it should require retention of their catalytic activity. Otherwise, it doesn't have any meaning to have a immobilized enzyme preparation. I mean, at the end of the immobilized preparation, retention of catalytic activity is a major requirement. Now, I think this is a very relative term. Retention of catalytic activity we must consider in a relative mode. Relative mode in the sense that 100% retention of catalytic activity at the end of any processing step is not feasible. So basically when we say retention of catalytic activity, we are implying a major fraction of the activity must be retained. Of course, the quantity of retention, the degree of retention will vary from enzyme to enzyme, as well as, I mean, the more unstable the enzyme, there will be poor retention. Or on the method of immobilization, more harsh is the method. That means if the enzyme is subjected to more harsh treatment in terms of pH, chemicals or temperature, the inactivation may be more, but ultimately there must be a major portion of enzyme activity must be retained. If the enzyme activity leads to, let us say, 0 or 5% or 10% at the end of immobilization, I don't think we should logically call it an immobilized enzyme preparation. The third issue, and which is of course operationally very important, is which can be used repeatedly or continuously in a process. Because ultimate objective of immobilization essentially is to use it either repeatedly in a batch mode, so that the same enzyme preparation is used, I mean, in repeated batches one after another, or it can be used in a continuous reactor like a plug flow reactor or a continuous stir tank reactor, so that you can process it, you can process the system, I mean, some substrate over a long period of time using the same amount of enzyme, and therefore decreasing the cost of enzyme per unit quantity of the product produced. In fact, the major objective ultimately, when we talk of the major objective from the industrial point of view of immobilized enzyme, would be to reduce the cost of enzyme input into any processing per unit quantity of the product to a significant level, and that is what is often achieved in the case of. So basically this definition essentially takes care of the three major factors. One is the localization of the enzyme or connectivity over a major substrate, and now such an immobilization can be achieved by different means. Of course, there are a variety of methods. Basically what we are talking of, that if you can couple, let us say, an enzyme preparation through a matrix, an insoluble matrix which is not soluble, and you can couple, whereas the enzyme activity is retained, and this being an insoluble matrix, a particulate matrix, one can pack it into a column, or one can use it in a stirred reactor and give a feed continuously and take out the product stream continuously. Both the things are feasible. Alternatively, instead of coupling to a matrix, it could also be feasible that you entrap the enzyme into a matrix. For example, you take, let us say, microcapsule, a permeabilized or a permeable microcapsule in which the enzyme molecule can be entrapped, such that the enzyme molecules cannot come out, but the substrate or the product can freely diffuse in and come out respectively, and such a system will also called immobilized system. Now, a very extreme situation can be considered of immobilization, where we are not really having a physical preparation in the immobilized form, but ultimately, for example, if you use an soluble enzyme, say for example in a membrane reactor, just a typical membrane reactor, and let us say this is a ultrafiltration reactor in which the enzyme molecules are put in, and this membrane is able to restrict the flow of enzyme, and the product and substrate, you can put in the substrate here, the product can go out, and the enzyme can be retained in the membrane reactor, let us say an ultrafiltration cell, in which the molecular weight cutoff of the membrane is such that the enzyme is not allowed to pass through and is retained as a retentate, whereas the substrate and the unconverted substrate and the product formed is passed through the membrane and can be collected. Now, in such a case also, as a system, we will call as a immobilized enzyme system, although the preparation will no longer be immobilized in the physical form, in the form of like what we consider as the bound preparation or a membrane entrapped preparation, but still functionally, because in such a system you can use the enzyme continuously or repeatedly without losing in the product stream. So therefore, all these systems will be considered in the format of immobilized enzyme preparation, and the enzyme preparation may be in the form of coupled form to a matrix, insoluble matrix. It could be entrapped into a semi-permeable either semi-permeable cell, or in the form of entrapped in a gel, because gel also provides you a porous network through which in which the enzyme can be entrapped, and they will not be allowed to pass out through the gel pores because of the size limitation once they are entrapped, and the similar thing applies to a ultrafiltration reactor. So basically, yes. You see, what I wanted to bring a point to your notice was that we often consider that if you couple an enzyme molecule, soluble enzyme molecule, onto a solid matrix, whether it is a inorganic matrix or an organic matrix or a gel, that is a conventional form of immobilization. Unconventionally, one can also make all these conditions feasible. That means you can localize the enzyme in a defined area of space, that is in a ultrafiltration cell, and I presume that you understand what an ultrafiltration cell is. That you have a stirred vessel, in which at the bottom there is a membrane with a narrow molecular weight cutoff. That means it will allow only substances below a certain molecular weight to pass through it. The molecules with the higher molecular weight will be retained. Now, in such a reactor, if you use an enzyme by putting substrate in and taking out the product, which are low molecular weight compounds, an enzyme being a high molecular weight molecule will be entrapped, will be retained in the reactor during use. Now, such systems also will be considered as immobilized enzyme systems.
Can you give some examples where this kind of interrelationship is used?
In fact, although industrially such systems are not very commonly employed, but as far as the study is concerned, academic study is concerned, these systems have been applied. In fact, one of the early systems which were applied were way back in late 60s and early 70s to hydrolysis of cellulose and starch, because starch product is low molecular weight, glucose. Similarly, cellulose product is also glucose, which comes out in the product, and the enzyme and the unconverted substrate remains in the retentate. So those are the systems where, and in fact it will be applied usually where the, I think one limitation will be that the product has to be a low molecular weight product, which can be easily, I mean,
Falling can be a major problem in this.
it is, that's why it is not industrially, it has not become very attractive. There are problems still as far as the cost of the membrane is concerned, the fouling operational problems, there are still problems. Now when we say that we have, let us say today in the literature you will find probably a few thousand methods of immobilization, not that all of them have been commercially viable, because commercial viability is an issue which is linked directly to economics, the process economics. So it does not restrict to, but what we are talking of, the basic definition of immobilized enzyme, that you can do in that matter. As a matter of fact, this is an artificial cell, as you can consider, let us say, a living cell itself. Living cell also, the enzyme is entrapped through a semi-permeable membrane, which does not allow the enzyme molecules to pass through, and some of the substrate and product molecules, some of the product molecules from the cell also are easily trapped off. So in a way, in a sense, of course the cell membrane is much more specific and has quite distinct feature compared to a ordinary ultrafiltration cell which only functions based on molecular weight. The cell membrane has still more specific functions and more elaborate functions than a, but here the function is only based on the molecular weight cutoff. Now I think the second point is that what are the major advantages or what are the major features, why should we immobilize enzymes? I think there are mainly two motivating factors. I would like to mention here, just to give you a brief about the historical perspectives. The whole concept of immobilization of enzymes started sometime mid 60s, not before that, because mainly for two reasons. One is that the scientists wanted to apply some of the enzymes for industrial processing, and obviously for industrial processing you need certain operational advantages. The major disadvantages in the case of enzymes, that means the restriction on the batch mode of processing, the stability, some of the physico-chemical properties in respect of pH optimum, temperature optimum, your substrate specificity, they could all be taken care for industrial processing. The second issue, the second motivating factor was, many people also wanted to study the enzymes in their natural habitat, or at least as close to as the natural habitat. See, when we talk all about the enzyme structure and functional aspects, we are missing one thing, that whatever studies have been made in those aspects, they have been made after isolating the enzyme from a cell. It has been delinked from its natural habitat, and essentially when we take a, let us say, a enzyme sample in a buffered medium, in a buffer, its natural structure function may not be identical as it happens in the living cell, and that has been recognized very well, because many of the enzymes, if you notice the half-life of an enzyme in a living cell, if you make certain indirect calculations, you will notice that in a living cell the enzyme is not as unstable as we notice it in the laboratory or for industrial application. The turnover of the enzyme molecule itself in a living cell is quite low. The cell does not synthesize, because protein synthesis in a living cell is quite an energy intensive process, and cells often will like to economize the energy consumption as far as the protein synthesis is concerned, and therefore the half-life of the enzyme in living cell, the stability is much higher compared to a free state as we get And so that was another factor. People thought that if they can try to bring as close to the living environment, and the process which they adopted was to immobilize, because most of the enzymes which people understood very well were the membrane bound enzymes, and they noticed that the membrane bound enzyme, there was some kind of a physical coupling between the enzyme and the insoluble membrane matrix, membrane as a matrix, and therefore they thought that coupling of an enzyme to an insoluble matrix would be a desirable feature to study enzymes or immobilized enzyme as a model system for natural enzymes. And of course then one can really look into the various properties in terms of their operational properties, in terms of their kinetic properties, and also in terms of their substrate specificity and other aspects. So these were the two. Yes.
Isn't the immobilized enzyme also purified and then coupled with the insoluble matrix?
Yes, but again after isolation from the cell, you are trying to bring back, because you see when they are in the living cell, you have no means to study them, excepting indirect calculation that okay, this might be happening, this is the flux, this is the rate of flux. In fact, most of the metabolic pathways, although we understand qualitatively much better, but still the quantitative fluxes of the individual enzyme catalyzed reactions in a metabolic pathway is still a very difficult aspect. So the only way out left to a scientist is that one isolates and tries to mimic the conditions of a cell, so that one can probably try to interpret the functioning in a cell, how it is happening. If you can mimic, and the immobilization provided, you isolate the enzyme, of course you don't keep it in the cell, you isolate, but then again revert back, let us say kind of a pseudo cell. For example, you entrap that into a microcapsule which is contained in a semi-permeable membrane, or take a very fine sort of film, a porous film, and attach an enzyme molecule to this, and then obviously this mimics as much as probably your enzyme was supported on a membrane in the living cell, and study its properties. So they were the two motivations. And of course when you talk about the operational advantages, I think from the point of view of our further study, I think because our major interest will be to look at enzymes as an industrial catalyst for use in industrial reactors for conversion of substrates into product. I think the immobilization offers a number of advantages, and of course one of the major advantages, as you would notice, and of course we have already talked about in the definition itself, is its reusability. That means the cost of the enzyme per unit product is significantly reduced by the reusability. Then you can also have an advantage, instead of being possibility of using only in a soluble mode in a batch reactor, you can use in a variety of modes, variety of application modes. A variety of reactor types can be used for carrying out the transformation or reactions, instead of using only a batch stirred reactor, as we can afford only for a soluble enzyme. And then you can, I think another major advantage is the rapid termination of reaction. So in the case of a soluble enzyme, the only means of terminating a reaction is to deactivate the enzyme, either by drastic change of pH, heating, boiling in a water bath, or adding some kind of a chemical which can inactivate, irreversible inhibitor of an enzyme which can inactivate the enzyme. But in the case of an immobilized enzyme, you can rapidly terminate by physically separating it, by simply separating, even if you use in a batch mode, by physically filtering off the enzyme preparation, the reaction is stopped. There is no enzyme present and the reaction is stopped. And that gives you a very great operational significance, and of course the use of a immobilized enzyme also leads to relatively purer product. You see, in the case of a batch mode, the enzyme is going along with the product stream, and obviously in some cases the discharge of proteins into the product stream may cause purification problems, and therefore the use of immobilized preparation in those cases will lead to a much purer product or significant convenience as far as downstream processing of the product stream is concerned. And in fact, many of the products which have been commercialized using immobilized enzyme preparations essentially are for application in, let us say, food, as a food constituent. Now in the case of food constituents, we don't want essentially an enzyme protein to go as a product in the product stream, and therefore in the case of immobilized enzyme that problem is not there, because the enzyme is already separated and the product stream can, as it is, be used as a food constituent in the directly as in the product form. Then of course in many cases, or rather in most cases I would say, the immobilization has also resulted in some altered physicochemical properties. When I say altered physicochemical properties, I think the most important property which probably for which enzymes are considered as poor candidates are the stability. In most cases stability is enhanced, and therefore it becomes a desirable feature. The other altered physicochemical properties are optimum pH and specificity. In many cases, of course, the altered optimum pH will result only on the basis of the nature of the carrier you are using. If it is a ionizable carrier, charge carrier, then only it will lead to a change in optimum pH, as we will see subsequently. But specificity also has been a factor. In fact, even one can have an altered Km for an immobilized enzyme as compared to soluble enzyme, again depending upon the same feature as optimum pH, that is the matrix is charged and the substrate is also charged. Because of partitioning effects, the apparent Km might also change. And the third motivating factor, as I mentioned, was the use of the model system for natural enzyme that we discussed, and which also has been a very significant reason for immobilizing enzymes. Now let us look at what are the requirements. Let us say when we talk about immobilized enzyme system, now what are the major requirements? There are, in fact, any immobilized enzyme system has three major constituents, three major players. The first player is the soluble enzyme. You have to start with a good preparation of an enzyme isolated from a living system, produced by fermentation or isolated from animals or plant cells. You need a soluble enzyme. Now, and the other is carrier. Now, you need a matrix to which the enzyme can be immobilized in general. Of course, there might be some methods, as I mentioned in the beginning, which may be carrier free, but in most cases carrier is a candidate which is required for coupling of an immobilized enzyme, a soluble enzyme onto an insoluble matrix. The third player is the method of immobilization, and the total performance of immobilized enzyme preparation will depend upon the combination of the interactions between these three players. Now, what are the properties we look at when we choose a soluble enzyme? Let us say we understand that we require a particular reaction to be catalyzed, so the enzyme is fixed. Take for example, I mean just as for the sake of convenience, let us assume that we have to isomerize glucose to fructose with an enzyme called glucose isomerase. Now, when we talk of the enzyme for immobilization, if we want to do this with the immobilized enzyme preparation, now what properties are important for choosing the enzyme source? Now, one is essentially a major factor is that we will look for an enzyme preparation which has a high specific activity. High specific activity means the catalytic power of the enzyme is much high, that is per unit weight of the protein its activity is much higher. So obviously this will depend upon two factors. One is the source of the enzyme and the purity of the enzyme. Purer the enzyme is better for immobilization, because you can have a very highly active immobilized enzyme preparation. Or the source will also be important, mainly if you can produce if you can choose a source which gives you an enzyme which has a much higher specific activity. That means your processing cost can be much lesser, that you have to produce much lesser protein for achieving the same catalytic function. Then desirable operational parameters. Now, one of the major constraints on operational parameters is, for many processes, that the operational parameters required for an enzymatic catalysis should not induce any side reactions. Any side reaction. Say for example, take again the example of glucose isomerization. Now this process, glucose isomerization, in addition to catalytic function by glucose enzyme, it can also take place in the presence of an alkali. If your environmental pH is alkaline 7 plus, the isomerization can take place naturally, and the isomerization product will not be only fructose, but it will be plus a few other hexoses which will be produced, and essentially the whole advantage of using an enzymatic process is lost. Now in such case, if you can have an enzyme preparation which has either a neutral or a slightly acidic pH optima, is desirable. And in fact such efforts can be, the enzyme immobilized enzyme preparation can be tailored, so have to have at least minor adjustment of the operational parameters. And of course the third and the another factor is stability. Again, in the light of only your specific activity, again this will also depend on source of the enzyme. So one needs to look at choose the enzyme and its further processing for purification, so I have to have the three properties which are in the positive enzyme, and we choose the soluble enzymes on that basis. The second candidate in the case of immobilized enzyme are carriers. And of course, in terms of the total gross weight of the product, for example, if you look at the total immobilized enzyme preparation, I think almost 99 point a few points will be contributed by carrier. In let us say 1 gram of immobilized enzyme, the enzyme preparation will be probably only a fraction of milligrams, or if more, not more than 1 or 2 milligrams. Bulk of the product, immobilized enzyme product, will be in the form of carrier, because ultimately you are taking a water insoluble carrier. I think one of the key parameter here is it has to be insoluble in reaction environment. Now I am using a general term reaction environment mainly because while most of the reactions are to be carried out in aqueous medium, today there are requirement that many of the reactions may be carried out in organic non-polar medium. So therefore the carrier which we choose should be insoluble in the reaction environment. That is a major requirement, and of course it will ultimately depend upon the chemical composition of the carrier. Of course logically the carrier should also be non-toxic to the enzymatic reaction. This should not act as an inhibitor or some kind of a irreversible inhibitor to the enzyme catalyzed reaction. Another very important feature is your resistance to microbial, thermal or mechanical degradation, because as you recall, right in our definition we mentioned that it is required to be used over a long period of time, and the use over a long period of time may involve many features which might expose the whole material to many features which may lead to microbial degradation, thermal degradation, or mechanical degradation, and the carrier should be resistant to them, and therefore it should be, carrier itself should be very stable, otherwise the stability of the enzyme will not have much of meaning. Then of course the carrier should also have a large number of sites for binding of enzyme, because the major reason is that ultimately the objective of an immobilized enzyme system will be to have a very high activity, to have a high functional reactivity. One need as much of enzyme, I mean the activity of the, or loading of the enzyme onto the carrier must be very high. The higher you can make, and that will require a large number of sites for binding of the enzyme onto the carrier, and of course this will again require many of the functional groups present on the carrier. It will also depend on the porosity of the carrier, and of course depending on the surface area, which will also result from the porosity as well as the particle size. So surface area will also become quite important. And finally, and if not a major scientific fact, but at least a economic feature is the regenerability and cost is another criteria which plays a very key role in choosing carrier matrix. See, normally we will prefer a carrier which is not very expensive. Of course, in an inexpensive carrier one may say that you may not be able to meet the other desirable properties. One can choose an expensive carrier provided the carrier can be regenerated at the end of the exhaustion of the enzyme. Because of course when I say immobilized enzyme should be used continuously or repeatedly, again this means that it can be used for a couple of days, couple of months, couple of years, not forever. At the end of the cycle of the operation of a immobilized enzyme preparation, if the carrier can be regenerated, it is desirable, even if its cost is high. In fact, as a matter of fact, you will notice some of the industrially used immobilized enzyme preparations, the low cost carriers have been rejected, mainly because of some of the undesirable properties, that they cannot get you a very high enzyme loading on them, and many of the expensive carriers have been used, but only under conditions when they can be regenerated. You see, carriers like ion exchange reagents, organic ion exchange reagents like DI cellulose, DI cephalodex, they have been extensively used for immobilization of enzyme by adsorption, and because even after the use of, when the whole enzyme has dissolved, the carrier is again available for reuse. And the final, now essentially therefore to take care of all these desirable features of carrier, the properties that you look in a carrier are essentially chemical composition, specific surface area, that means surface area per unit weight of the carrier, particle size, and of course ratio of hydrophobic to hydrophilic groups, mainly to achieve a very high degree of loading. Because one has to choose some of the functional group of the enzyme molecule for coupling, therefore
Ratio of
hydrophobic to hydrophilic groups on the carrier. And finally the cost. So these are the four general features or properties one has to look in the carriers, a variety of carriers that are used, and of course as you will notice that a large variety of carriers have been used and are available for use, fortunately, and these carriers have been classified by various people in different modes, in different forms. Now a very generalized scientific approach to classify them will be on the basis of their porosity, porosity as a basis for classification of carriers. That is, they can be non-porous. Of course, as you will notice that the non-porous carriers will essentially will have a low specific surface area, because a porous matrix will provide lot of internal surface in a matrix, which will not be available in the case of the only surface which is available is the external surface on the part, and to get a higher surface area you have to reduce the particle size, which also has a great limitation of use, because of smaller the particle size, its use in a packed bed is difficult because of the high pressure drops. Its use in CSTR is difficult, mainly because of its loss through the product stream if it is too fine a particle, and therefore non-porous matrices usually have a limitation of low surface area. Of course, as far as the chemical composition and ratio of hydrophobic to hydrophilic group or the cost is concerned, is general feature, but again non-porous matrices also has an advantage. Mainly they have been used extensively for biomedical applications, where a desirable feature, an additional desirable feature other than the ones we mentioned in the carriers, is the prevention of interfacial immunological responses in the biomedical system. For example, when you want to use a immobilized enzyme preparation for any biomedical application, it will interact with the bloodstream, and very often any external material in contact with the bloodstream will generate a very vigorous immunological response, and obviously it has been noted that the non-porous matrices or the non-porous materials are much softer as far as these responses are concerned compared to porous ones. And a typical example of use of the non-porous matrix in the biomedical application has been nylon as a non-porous matrix, which has been used to immobilize some of the therapeutic enzymes for use in the in contact with the bloodstream. Then the other group of matrices is porous matrices. Of course, the most preferable kind of materials that are used for industrial applications, and essentially these matrices are controlled pore glass, controlled pore titanium, and a variety of other synthetic rather tailored materials which have been produced commercially for generating high porosity for coupling of enzymes on them. The third kind of material are gel structure, which essentially mean they are porous in nature, but they are usually flexible in the pore size, or they are more elastic as a matter of fact, and the porosity is flexible, and the typical examples are, which have been used, collagen, polyacrylamide, gelatin, and so on. Of course, a large number of other gel structure have been used. Now in the gel structure you can entrap the enzyme, you can entrap in the gel matrices, and the advantage is that the enzyme can experience almost the same kind of water activity or the environment as very close to what is obtained in the case of a living cell, and also will not the gel matrix, let us say if you consider the gel matrix and the enzyme entrapped in the lattices of the gel, will not allow the enzyme to pass through, but the small molecular weight substrate and product can easily be allowed to come out, get in or come out. Of course, another basis of classifying the carriers will be on the basis of chemical nature. One can have either inorganic carriers, yes, inorganic carriers or organic carriers. In most cases, the inorganic carriers, as I mentioned, are the whole range of control pore glass, control pore titanium, many of the clays which are naturally occurring like bentonite, animal charcoal, things like that, they form under inorganic, but the organic carriers are usually polysaccharides, proteins and synthetic polymers. Typical example of organic carriers are polysaccharides, your cellulose, agarose, even cross-linked dextran, what we call as industrially as Cefadex. This is nothing else but cross-linked dextran molecules. Then in the case of proteins, almost like what we talked about, gel structure collagen and gelatin are typical example, and of course there is a whole range of synthetic polymers which have been reported and used for immobilizing enzymes, which are usually polystyrene based, vinyl polymers, polyacrylamide and so on. So basically you can classify, and a large variety of carriers are available for use for immobilizing enzymes, and the choice will depend upon various properties that we have listed.